see. Okay, are you all able to see the presentation full screen? Okay, excellent. Thank you very much. So good morning, afternoon, or evening, depending on where you're joining us from. My name is Aleksandra Foksinska, and I am the program coordinator for the UAB Hugh Kaul Precision Medicine Institute at the University of Alabama at Birmingham. And today I'll be presenting on artificial intelligence-driven drug repurposing for rare disease. As an overview of my presentation, I'll first introduce the role of artificial intelligence, or AI, in biomedical knowledge. Then I will explain how my group's AI tool, Aleksandra, works, and briefly explain our review process and provide kind of two cases as examples. And then finally, I'll go over some of our initial kind of beta test results for Project 8P and touch on next steps in our collaboration with Project 8P. So you might be asking, why AI in biomedical knowledge? So first and foremost, approximately 2 million new scientific papers are being published each year. And this is an insane amount of information, and I have yet to meet anyone who's read every single paper out there. It's also kind of impossible to keep up with, which brings me to my second point, that over time, information and knowledge can tend to be forgotten, especially with these new articles coming out every single day. And also uninferred. And I know that is not an actual word, but what I mean is that there are relationships and conclusions that can be drawn between pieces of knowledge that aren't, since it would take someone reading all of these papers and inferring all of these relationships. And then lastly, getting the knowledge contained in these papers, as well as information and data contained in data sets and databases online, into a searchable format that is also user-friendly is a major obstacle. And so these 3 points are where AI helps us out. So how does it work? So first we start off with knowledge. So imagine each of these dots is a concept, and each color might represent a different category of concepts. To first even attempt to understand how these concepts are related, you need to have some structure or organization. And once you have some structure, then you might be able to start seeing relationships forming. And that's when you start creating knowledge, when you start linking these, whoops, a little bit of feedback there. When you start linking these different concepts together, you start building this knowledge. And each piece of knowledge is pretty unique. And for kind of, it could represent like a general relationship. So like in this example, the concept represented by the green dot, it interacts with the concept represented by this gold or gray dot. And the relationship could also be directional or more specific. So it could indicate that a concept inhibits another concept. And also, all of this kind of structured form of knowledge is also known as a knowledge graph. But if we would like to see a little bit more complex relationships, like for example, if we started off with this concept and then saw this relationship between this one, and also this third concept, we could potentially stop right there and think, well, maybe that third concept is somewhat related back to that first concept. Of course, this is not direct, but by the other relationships established in this graph, you could kind of arrive at it or infer kind of this indirect relationship. And the same thing goes for example, for this fourth concept. It can also be indirectly linked back to that first concept. And this is what something called a knowledge reasoner does. And this is what Aleksandra is, a knowledge reasoner. And these reasoners kind of perform more of this complex thinking. And I know this isn't a human, so can't really think, but it can establish or kind of give us some inferred relationships. And you might be thinking, you know, all of these dots and lines, what does that have to do with biomedical knowledge? Well, if we isolate this particular example and then turn each dot back into, you know, the concept like I was mentioning, we could see that there's this relationship between this disorder and that it disrupts this particular pathway. And then we have this gene that's associated with the same pathway. And then we have this drug that activates this gene. And each solid line and arrow, I guess, would be representing a direct known relationship that has some evidence behind it. And evidence could be like a publication that establishes the relationship between this disorder and this pathway, this gene pathway, or the drug gene relationship. However, you can reason or infer using like knowledge reasoners that, for example, this gene may be related to this disorder, or kind of similar. This drug may impact this pathway, or perhaps this disorder even. And so if a human were to try to infer these latter relationships, the ones that are dotted lines, they would probably have to read 3 or more papers to kind of arrive at this. And so knowledge reasoners help us out by trying, and I kind of emphasize trying because they're not perfect by any means. They try to help us out and kind of bring to our attention these uninferred connections. So what do you need to run a Metacanon search? So our process, we always start with a gene, and we've gotten pretty good at monogenic or disorders that are associated with a single gene, but often you have a group of genes or multiple genes, like for Project 8B, that you want to take a look at. Then it's also important to keep in mind the direction you would like to focus on in terms of regulating the gene or the protein. So sometimes you might find yourself needing to activate or increase activity, or perhaps decrease or downregulate or inhibit certain protein or gene activity. And then finally, we have to manually review any Metacanon hits that are returned to us. And like I said, AI is not perfect. And so sometimes, for example, a gene may have an older or less commonly used name that the AI, just because it wants to be thorough and comprehensive kind of in its search, it includes it in the search. And so sometimes you have situations such as this one, and this one's a real example that I've personally seen, is that I was looking for information on the gene CMAS. And at some point, CMAS gene also was known as CSS. However, CSS has been used in some publications to stand for cesarean sections. And so you can kind of get some of these false positive hits. And so that's why we do a manual review process to ensure anything we're returning back to a clinician or researcher is a true hit. As for what types of hits we get, so if we have a gene target, sometimes we get lucky and we find the drug that is predicted to directly regulate a gene target. So we call this a direct or one-hop query because you kind of just hop one step over. However, what if there's a gene or pathway that regulates your gene target of interest? Well, we could take a look at drugs that regulates that gene or pathway. And so this is more of an indirect, or we call it a two-hop, because you kind of hop twice over back to your kind of original target. And so once we have drug hits to take a look at, you may be wondering if they're ranked or prioritized in any way. And The short answer is yes. Medicanerin uses publications to rank drugs. So the more publications Medicanerin pulls out that reference the drug interacting with the gene target, the higher it appears on our list. So now I'm going to provide two kind of case examples of Medicanerin in action. So in this first case, we have a gene of interest, ADNP, and the disorder associated with ADNP is kind of a deficiency in ADNP protein in the body. So there's not enough of this protein being produced, and therefore there's an interest in upregulating ADNP. And we put it through Medicanerin and found a drug. Particular drug is ketamine. And this is not something most physicians would, not think twice about. So obviously any results we do get that may be of interest are first vetted by our own clinical team. And then, excuse me, and then definitely before, anything gets back to the patient, it is vetted by researchers, the patient's own clinical team, and so on. And there's many discussions and meetings kind of happening in the background. But ultimately, ketamine was used, or it is being used in a clinical trial at Mount Sinai for ADNP syndrome. And we don't have the official results yet, but we have heard that so far the results have been encouraging. And then in another case, this one actually being COVID-19, we had a very long list of potential genes that were associated with increased or decreased susceptibility to SARS-CoV-2 infection. And one of these, TMPRSS2, this gene, was recognized to be important in getting the virus into the cell. And so although we didn't find any drugs that directly inhibit or downregulate TMPRSS2, Medicanerin did bring to our attention that androgens actually induce expression of TMPRSS2. And so that kind of became I guess, another target of interest for us. So we took a look at what decreases kind of androgens. And there's a drug, Aleksandra, that did just that. And again, we, passed along our results to researchers and clinicians. And after many discussions, actually the VIA decided to do a clinical trial. And the results came out earlier this month. And although there wasn't a significant difference between the treatment and control groups, which could be attributed to many different things. And this paper does a great job of listing out some of the differences between this trial and also other trials that looked at androgen suppression or kind of androgen receptor antagonism and had some more positive, significant results. This still kind of provides a powerful example of what AI can do. So now I'm going to try to kind of tie in everything I just covered with our preliminary or kind of beta testing results for Project AP. I think everyone here on the call knows by now that there are a lot of genes on the P arm of chromosome 8. And it's about 250 to be exact. And many of these are deleted or duplicated based on each individual's genetic makeup. So one key element in question that needs addressing is how to prioritize this gene list and which genes could be kind of attributing more so than others to the overall phenotype. And some of this work has been started and definitely addressed, for example, in this awesome publication, and also work done by Sam and the Polarity group that who combine kind of deep learning on symptom and gene relationships and other publication mining tools, as well as in-field expertise with a focus on druggability, produced kind of 7 top gene candidates for initial Medicanron searches. So we took this list of 7 genes. The first 5 of these genes are commonly deleted in the patient group, and then the other 2 are commonly duplicated. And as for direction, it would make logical sense to upregulate or increase what has been deleted, and then the opposite for the genes that have been duplicated. You would want to, or you might think, to downregulate or decrease. And before I get kind of ahead of myself, I want to say that generally it's easier to create drugs and find drugs that inhibit versus drugs that activate. And kind of the best and simplest explanation I can give is that if you think about, like if you gave a child a toy, there are many ways that they can break the toy. They can throw it down the stairs, they can put it in water, they can cut something off, they can leave it outside, but there are much fewer ways that you can cause the toy to work better, or just even harder to fix it after the damage has been done. And so when I show you our results in a few slides, don't be discouraged that there weren't as many or any hits for the genes that needed to be upregulated or increased. But kind of continuing on, when we did run the list of these 7 genes, we had 500 results, which was very exciting. However, like I said, we have to manually review all 500 of these hits. And this review was carried out by one of our awesome undergraduate students, Elena, and she went through to sort out the kind of true positive results from the false positive results. And here's what we found. So kind of not surprisingly, the only target that yielded direct drug hits was CHRNA2, which is one of the genes that is typically duplicated, and you see kind of, and I guess we would want to downregulate it. But there were 3 other genes that had some promising indirect drug hits. However, none of these hits are the type or have enough evidence to suggest they should be tried immediately, or used, put forth in a clinical trial or anything like that. However, these hits could be interesting to perhaps test in an animal or cell model to verify their intended action. And finally, we will definitely continue our collaboration with Project 8P and Prolara, with some next steps being refining the list of candidate and priority genes, running new Medicanron searches and reviewing those results, and then passing along top drug candidates to be tested in models. So thank you everyone for listening, and I'm happy to take any questions at the moment. I think I see a few that have popped up in the chat.
And I also want to give a chance for Travis to chime in here as well in terms of the gene prioritization, the work that Prolara had done with Sam. I know that sort of was a complicated process condensed into one slide, so definitely among our researcher colleagues here and friends, if you wanted to get some more color on that, we can definitely explain that more. But we also want to balance some of the questions that will come from the family side that may not be as much focused on that level of detail at the moment. But yeah, let me take a look at the chat first here. So please drop in. But Travis, maybe you want to?
Yeah. Thank you so much, Aleksandra, for on short notice kind of presenting our slide. And it was a very thoughtful process involving a lot of different data streams. The idea being that because we were looking through literature for potential drug targets, for drugs that we could potentially repurpose for our targets, we wanted to use at least a couple data sources from that, as well as information from our experts like Dr. Chung about what they think may be the most causative agents along the chromosome. So it was a very thoughtful process. I think, Aleksandra, just one point of clarification that I should have made more clear in the slide that I sent you is druggability is something obviously that we think we wanted to think a lot about for the use for Medicarin, but we actually didn't use for gene selection this round, right? So it's something that we should think about, and especially moving forward, it's something we want to consider. But for this round, we really just focused on genes that rose kind of bubble to the top in terms of potential causality instead of druggability, which, as you had mentioned, led to a few more deletions, which are harder to target with drugs generally than amplification. So that was just one point of clarification, but also a point of optimism, right? As moving forward, we could potentially kind of better prioritize druggable targets for future things with Medicarin.
Yes, absolutely. Thank you for clarifying that.
All right, so I'd love to open up for questions. I know we have two different sort of audiences here, maybe with different level of detail, requiring different level of detail, but please, if someone wants to break the ice on either the researcher side or the family side, if you're a researcher, please just introduce yourself. Sorry for not giving everyone a thorough intro, but figured when people came up to speak, they would be able to summarize sort of who they are and what they're going to be working on, and then what their question is. So please, anybody, the water is open.
I will get started on this. Thank you, Aleksandra, for your talk. Thank you, Ethan, for organizing, and everyone for attending. Obviously, it's critical to get this list of genes as close as possible to, what is most significant. And I was wondering whether from the 250 genes, did you have the chance to select perhaps genes that individually being mutated are causing a disease?
That's a perfect
some of
question.
Of them will be involved in other diseases.
Yes,
and
so
perhaps a different combination.
Exactly. So that was actually, Aleksandra mentioned 2 machine learning approaches using the literature, and that second one with the name Amelie, that specifically looks for Mendelian inherited genetic disorders associated with the gene that are linked to a phenotype that we see, or a symptom that we see frequently in 8P. So the idea is combing through the entire literature, published literature, to look for Mendelian disorders that are disorders that are strongly inherited based on a single gene associated with a symptom or condition that we see frequently in 8P. So that's exactly kind of what the Amelie was looking for to help us prioritize genes in that list. Of course, not all the genes that showed up have a direct phenotype, something that is exactly what we see in 8P, but because of the complex chromosomal changes we see in 8P, we may not necessarily expect that, right? So again, it's a complicated, it's not a one-to-one matching, but that was the idea of using the Amelie, is to look for, okay, in the published literature, are there genes that cause a condition in and of it themselves, right, without any other mutations or abnormalities around them. And that was used as part of the choice for these original 7.
Yeah, and the other thing is that, so it's unfortunate that some of these are probably not in the database as druggable or being already put on the pathway to be for drug discovery. But in terms of phenotype for their downregulation or upregulation, I guess that's a different aspect, and perhaps just verifying that gene, when it's up or down, has a cellular or organ phenotype in whatever model. It's perhaps another line of information which will enhance the information that is relevant, or perhaps can be put on the side and something else can be brought up.
Yes, absolutely. Definitely dosage sensitivity. Some genes, you can have a deleted copy and be okay. Whereas some, then you're stuck with a deficiency or haploinsufficiency. And then same with kind of duplication regions, that some genes are triplosensitive. So having 3 copies is not good and will lead to a phenotype, but some may not. So that is a very good point, that kind of the dosage, I guess, sensitivity of these genes is also an important aspect to take a look at and keep in mind when prioritizing genes.
Thank you.
Thank you.
Thank you, Nicoletta. Anyone else would like to have a question or make a comment? Faye, you've raised your hand very politely. You're next.
I'm being British tonight. So can I just ask about your drug database? And like how big is it? Like, exactly, does it just sort of like active site? I don't have a concept, like, and that was, by the way, that was an absolutely beautiful presentation. I just like, wow. But I didn't kind of get a concept of, when it's sort of, crunching the data, what exactly it's looking at in terms of active site of protein and, active site of drug. If you could talk us through that, that'd be brilliant.
Yes, absolutely. And it's a combination of different things. So Medicanron, being a knowledge reasoner, is reliant on the knowledge graphs that are imported into it. And so the knowledge graphs that Medicanron currently utilizes, they're all, I believe, from different teams that are working with the NIH and CATS, or the Center for Advancing Translational Sciences. And so some of these knowledge graphs, many of them are just, for example, there's one, I believe is SEMED. It's a knowledge graph that takes information that is contained in abstracts of publications. And so it reads like all the available abstracts and creates these kind of knowledge graphs based off of the information contained in abstracts of papers. And so a lot of our kind of review process is actually looking at abstracts from papers and seeing if the connection is correct. So, is this the correct gene? And, is this drug actually regulating this gene in the correct direction that we want it to? That's part of our review process. So most of our hits are actually taking literature evidence in the form of abstracts, which also limits kind of the information we are looking at. However, there are teams working to try to incorporate more full text publications and therefore increase the amount of potential hits that can be generated. Even some teams looking at how to extract information from like the figures contained in publications or supplementary materials so that there's a better chance of possibly getting something out of that. But there are also other knowledge graphs that are trying to incorporate information contained in like drug bank and I believe like the L1000. There's the different kind of databases there, but those are slowly kind of trying to be incorporated. However, it's always kind of a hit or miss with those because you can throw in a gene and it spits back a lot of drugs. However, it was like drugs found to upregulate this particular gene amidst all other, hundreds of genes that were studied within a certain cell line, which may or may not be applicable depending on the specific cell line. So I think that was a very long way of answering your question, but I hope that kind of clarified how some of these drug hits come about, most of them being from publications, specifically abstracts of publications.
Yeah, no, that's absolutely brilliant. And I think as well, it kind of, in my mind, and correct me if I'm wrong, just says to me as well that like, there's a lot more still to be explored.
Yes, absolutely. So there's so many teams kind of working on how to get information that's already out there and get it into, a good searchable format that's also user friendly so that, people like me can use it, but also clinicians, researchers, can go on and find information, and not just drug information, but also, it's it would come in handy also when you're trying to help diagnose someone, and you have some different variants and you're trying to see disease relationships and things like that. So one thing is, drug repurposing, great, definitely for rare diseases and other diseases, but there's also so much more that you can do with AI in these kind of knowledge sets that have been created.
Sure. Thank you.
Absolutely.
Ina, you're up next.
Okay, wonderful. Thanks, everyone. So Aleksandra, is the UAB team, like when you say these teams are working on it, is it outside of UAB or internally to what you guys are trying to,
Yes.
build, like accessing the figures, the data hidden in the supplementals, and then kind of adjacent to that question for compounds, do you also try to access the list of compounds that were not approved by different regulatory bodies?
Yes. So first question, outside of UAB, who else is there? So I believe we're one of like 15 groups. So kind of like there's the UAB group with Medicanron, but we're like only one little part of the whole kind of translator program. So the NIH NCATS has a translator, biomedical translator program. And there's different kind of teams working on it, and they're all over the US. And there's some that are dedicated to building knowledge graphs that then reasoners like Medicanron can utilize and kind of be the interface between the user and the knowledge and the information that's there. So there are other teams kind of taking a look at it. There are teams that kind of specialize in the different data sets that they're trying to kind of incorporate. So there's some groups that are very heavy kind of cancer focus, and they use cancer kind of examples, tissue samples, kind of transcriptomics, I guess, that you can run on cancer tissues. And they're trying to take that information and get it into a form that could be useful and utilized by clinicians and researchers. And so that's the kind of whole translator, that whole NIH biomedical translator program, and Medicanron is just a kind of small part of it. However, it's all very, everyone kind of tries to work together. So we sometimes we have individuals joining us during our case reviews for the Precision Medicine Institute. So that's when we're kind of reviewing cases, and these computer scientists, most of them being computer scientists, join us on the calls, and they kind of see how we work through cases and run Medicanron. And some of them have actually gone back to their teams and they're like, hey, there are some pretty easy changes we can make to how we have this data, that could be useful for like the UAB team to run more of these searches for rare disorders. So they kind of see our process and then they try to build the tools to kind of help us. And so it's been very exciting seeing some of the collaboration between our groups. And then for your second question regarding just drug kind of, approval, Medicanron returns all drugs. So whether that is a drug or compound that is, right now just being used in a lab and probably toxic and not going to be developed for human consumption anytime soon, to FDA approved drugs, and also other drugs that maybe not FDA approved, maybe approved in Europe or somewhere else. That is something that they're trying to work on. Also through the NIH translator program, is to create kind of a web version actually of Medicanron, something that users can see the results, but also click on specific compounds and drugs, and they get kind of a snapshot of, this is an FDA approved drug, or maybe it's not FDA approved, maybe it's a supplement or a nutraceutical that might be of interest. And so Right now we do that kind of manually, and that's why we also have some physicians working with us, because logically we're like, oh wow, this compound, upregulated this gene in this model, that's great. And then the physician looks at the compound and they're like, yeah, this is not an FDA approved drug or compound. I'm not able to prescribe it to anyone. So, we either have to test it in an animal first, or we have to kind of go back to the drawing board. So did that kind of answer your question?
So thank you. And sometimes I ask these questions and I know your team well. And so I am asking for the interest of the group at large that may not be aware of that. My final question is for the group, and then Stefan, I do see your hand up. But it's for some of the other researchers that are listening to the call, like, how can these bioinformatic tools or even Travis's work that's supporting us through Prolara support some of the future data that we'll get from, the researchers that are on this call that I know are just getting started with us?
Yes.
Maybe, yeah. Oh, sorry. Go ahead, Aleksandra.
Oh, no. I was, it's okay.
Cool. Well, I was remiss earlier to not have our AP researchers introduce themselves. So maybe I can rectify that. I know, Nicoletta, you already spoke, but Stefan, you have your hand up. Maybe you could introduce yourself and then your question, and then I'll make sure we go through the other researchers here to make sure they get a chance to introduce themselves and then what they're working on, where they're located, because I know you've been sitting here patiently listening. So Stefan, go for it.
Okay, I'll try to be really short. So I'm Stefan Pinter. I'm an assistant professor at the University of Connecticut, and I work on trisomy 21 and Turner syndrome. So Turner and Down syndromes. And so my question was, the difference between flipping. I mean, between a deleted gene and a duplicated gene impacting its expression is ultimately down to flipping a sign in your knowledge graph, right? Because if you have, I'm prefacing this question more for, I know you know this, I'm prefacing this more for the patient families, but when you have a deleted gene, you don't have the right dosage of that gene, then a protein complex that requires that protein is going to be forming less efficiently. And so it's going to be falling apart. So it's going to be other genes that are also going to be down. But one way to attack those would be the same way by targeting a counteracting protein complex that now might be out of balance. So is your knowledge graph taking into account proteomics, protein interaction data?
Yes, and I guess I don't know how to describe to what extent, but I do know that anytime I do search a gene, usually in my results, I might get drug compounds, but I also might get other genes. And sometimes it's genes that regulate it. So sometimes I get transcription factors back. So these are, I guess, proteins that influence how a gene is being transcribed and expressed. But I also get interactors. So if you're familiar with StrainDB, where it kind of shows the different interactions, that's also kind of incorporated in Medicanron. So I can see some of these interactors, whether that's kind of upstream or something that regulates the gene itself, or downstream. So something that the gene is, part of a complex, like you mentioned with, or something else kind of part of the same pathway. And so oftentimes when we kind of hit a wall with, a single gene target, we try to see, okay, is there anything else that we know about this particular pathway that's being disrupted that we can go after? And like I mentioned, sometimes it's hard to enhance the activity or increase the activity of a protein. However, if down the pathway there's something that's being overactivated as a result of something, there's not being not enough of that protein or decreased activity of a protein, then we might go after that target. And so that's kind of like the indirect approach that I mentioned. So there is kind of an aspect of proteomics. I can't say for certain like how vast that is. That would be, I guess, better answered by the other teams that work on the knowledge graphs that bring in this information. But yes, there is other genes and complex information and other proteins that are associated with the gene target of interest.
Okay, and the other question that I had was, you could end up with counterintuitive results, right? Like seeing that a gene is upregulated with a certain drug may actually reflect that there's now more reliance on that gene product to actually, for example, for the cell to survive. And so that would be exactly the wrong drug to use in this case, right? I suppose you could run this knowledge graph backwards, right? Like you could actually start from your drugs that you get out, if you have at least, I don't know, a handful of drugs, and work backwards through the network to see how good am I actually at hitting back at those genes. Do you do those kind of bootstrapping tests?
Yes. So that is, there's actually like two ways that we run Medikern queries. One is kind of working, I guess the question is like, what regulates X? But we can also see what X regulates, kind of going back. But so kind of to answer your question, yes, sometimes we, have a particular drug or compound. And sometimes, I guess researchers get back to us and maybe they've done like an initial drug screen, just they picked out a few drugs that they had and they kind of tested it. And they're like, oh, we see, an improvement or, whether it's an animal model or a cell model, with this particular drug, are you able to tie it back to like the mechanism of, this gene and this disorder? And yes, there have been instances that we've been kind of able to work backwards, take the drug and kind of get back to a gene, or, some pathway or something that then kind of, I guess, branches off into additional targets and other drugs that could be considered. So that's, I guess we don't routinely do that with every case. However, we've done it many of times where we're interested in how well, can we track the drug back to this gene? Or if, we start off with a drug, can we track down a pathway and kind of meet in the middle between, what we know about a disorder and then this drug that, worked well in an animal or cell model, and then kind of get to the middle and try to see, what is the connection and reasoning, I guess, that uninferred connection that I kind of mentioned at the very beginning. So very good points. And yes, Medikern is able to do that.
Thank you.
Thank you so much. Nicoletta, you were the speaker last time, so I think people don't remember. You can maybe quickly introduce yourself, and then maybe you can turn to Jason, Aria, Hirui, if you had any questions, and then on the family side as well. I see you, Basil, you've asked questions before. I don't want to pick on people, but definitely we've got about 10 minutes left here. I'd love to fill it with some discussion.
So hi again, I'm Nicoletta Moisoe. I'm Associate Professor in Pharmacology in Leicester, United Kingdom. My part in the project is to look at mitochondrial nucleus communication in chromosome 8p disorder and addressing Bina's question on how this work that Aleksandra presented would be feeding back into what we are doing. Is that perhaps first thing that I'm going to do, I'm taking this list of genes and see whether themselves or some of their interactors may impact on mitochondria and the nucleus aspects of the interaction I'm looking for, like DNA damage, mitochondrial function and dysfunction. Some of these pathways, are they likely to be affected? And this is why I have asked about the monogenic aspect of the gene sets, because I'm aware that some of those genes have been linked already with mitochondrial dysfunction and DNA damage accumulation. So these are the links I'm looking for in these data sets. Thank you.
Thank you. Aria, Hirui, or Jason, any thoughts from your end?
Hi, I'll just introduce myself and say a little bit. So I'm Aryeh Warmflash. I'm a associate professor at Rice. My lab works on modeling early human development using models we build from human embryonic stem cells. To answer Bina's question, I think we're quite interested in these gene lists because we do a lot of sort of knockouts and things to understand the effect of knocking out genes in our models. So to the extent that we can prioritize those are very laborious experiments. And so I think both prioritize in terms of what is known to have an effect and known to have an effect when you only lose one copy, and also prioritize in terms of, well, if we were to find something, what would actually be actionable because we know there are drugs that target it. So I think we're interested in that from both ends. I was curious whether there are any sort of statistics available for your pipelines, like what fraction of the things that the computer pulls up actually make it through the manual screening, and then if it's maybe not enough has been done, but what fraction of those things make it to various endpoints in clinical trials. I think it'd be quite interesting. And also, is there any sort of feedback process where you say, okay, you know, the algorithm picked this thing out, but it didn't work, and we think this is why, and this is how we can make it better?
Yes, and you bring up a very good point, and that's something that we're kind of in the progress of trying to do something like that. I can't speak on behalf of, I guess, the Medicanron team. Like, I am not a computer scientist. I am a user of Medicanron. However, the team that actually kind of programs and runs Medicanron and continuously tries to improve it. I know that they have certain metrics that they're trying to measure of, you know, out of the results, like you mentioned, out of the results that the user gets back, how many are the kind of true positive hits versus the false positive hits or anything like that. We don't have the statistics right now of you know, I guess our review process and how successful it is or how many results we're able to get from it. But I do know kind of from the more clinical aspects, kind of our work with our participants, we're trying to get better. And we've been doing this only for about 2 or 3 years now, but trying to start collecting data on if we do get results back, any results that we do get, we create like a report that goes directly to a treating physician first. We never disclose any information to patients just because we are a research kind of consultation service and we can't cross that line and practice medicine. But these reports are sent to a physician, and oftentimes, you know, the physician, and it's usually a therapeutic or an option that's fairly safe and has been kind of vetted by the physicians on our team who say, you know, if this was my patient, I would be okay with kind of trying this and monitoring, of course, and everything like that. So we, you know, pass along our results to the individual's physician, and they get to review the report. They get to ask us any questions, and sometimes they decide to kind of move forward and try something. And so for one case, that was something as I guess, as small as a vitamin A kind of supplement. But then again, you definitely have to have a physician, you know, guiding any type of treatment, because with vitamin A, you can also have vitamin A toxicity if you take too much. So that is why we also route everything through a physician first. But with these reports that we're sending off, we've been slowly trying to collect data since we just got, well, just a few months ago, we got our IRB approved to actually start kind of gathering data from these reports that we send off and kind of the outcomes of those reports, and seeing if we can measure, you know, how well can Medicanron come up with a potential treatment option for different individuals with different rare disorders, and also kind of tracking outcomes, whether that's looking at like seizure frequency or kind of speech development and things like that, trying to see if there's any way we could track that and have some kind of measurable outcomes to show that, yes, there's been a significant change or difference because of this treatment option.
Thanks.
And Harui, any thoughts from you? I see Jason had to leave, and I know we've got about 5 minutes left, and I wanted to do a quick program note about next month's talk. But Harui, stage is yours.
Hi, I'm Harui. I'm an assistant professor at UC San Diego. Our lab is interested in understanding the genomics of neurodevelopment and cognition. We're in partnership with AP, are beginning to utilize induced pluripotent stem cells to generate the different neuronal subtypes and generate large data sets, including RNA sequencing, 3D genome architecture, and epigenome. I guess for us, what we've been finding with our studies with Down syndrome and other disorders that have genomic imbalance is that there's this genome-wide disruption of, let's say, the transcriptome, and we think that these data sets might actually provide a wider scope of gene sets that might be used for targeting through your system. And I guess this would be in partnership with both Parlara and your team. But we hope to generate some of this data and try to see how we can partner. But your data looks really great. I'm excited to start using it.
Oh, thank you. And we have worked with other groups that have done transcriptomics. Our kind of transcriptomic pipeline is still kind of in its early stages, but we've had at least one case that, a group has returned kind of transcriptomic profiling from a patient's, small patient kind of cohort. To see if we can, try to help prioritize the genes and with the genes that kind of end up, I guess, top candidates, seeing if there's any way to modulate those genes with a drug or a compound. So yes, definitely excited to hear that transcriptomics is part of the project here.
And any questions from the family side? It's okay if not. We definitely, you could follow up by email if that feels better, or this will be available for replay and for rewatching, and you can definitely follow up at any time if you so choose. I think with that then, with about 2 minutes left, I appreciate everybody hanging out for the hour. And I definitely appreciate Aleksandra for sharing these, sharing what all the activities are that are going on at UAB, and then these early results for AP, and how we could refine them and see if there's something to chase down. So really appreciate everyone also who asked questions and created I think a great flow today's conversation, especially the researchers for really engaging and asking, I think, all the right questions. So with that, I want to just preview that next month's roundtable will feature Professor Dennis Lal, who is building an online data platform, and he's worked on a number of CNV and neurodevelopmental disorders and part of the commission, and is hopefully going to provide a data infrastructure, or excuse me, I should say an architecture online to sort of store all the data and all the data sets that we want to generate and all the projects that have been sponsored so far. And so we're really excited to have Professor Lal join us and share updates on this online data portal for AP, and welcome everybody here to engage on that topic. So with that, I guess, Bina, any parting words or program notes from your perspective?
No, I just want you guys to all know that, like, sometimes it's not relevant to each of your, work in your labs, or maybe not aligned in the way that you kind of think about some of the translational work that we want to set ourselves up for. I'm speaking to the researchers, but I really do appreciate your time, and the community and the families do, and that's really important, because without the family engagement, none of this happens. So thank you for just having faith and believing as we continue to build momentum on these roundtables. And we really value each of your time. And thank you to all the families for just being on these calls and asking us questions on emails, and however you feel comfortable, please continue doing that. I mean, Ethan knows I ask very simple-minded questions all the time, and I'm constantly learning myself. And so again, many thanks.
Great. I just shared my email here as well, and Whitney's email. Please follow up with one or either of us if when it comes down to either the recording or trying to get connected with any of the researchers here. We're going to hopefully build some ways to collect feedback and have continuity of feedback and closing the loop on questions and stuff. But bear with us as we start to build out that infrastructure. But in the meantime, just email Whitney or I. That's the best solution here for getting connected to anybody or for chasing down any questions or getting answers to your questions. So thank you again for your attendance, and we look forward to seeing you at the next roundtable in July.
Thank you, everyone.
Thanks, everybody.